Millions of microorganisms are present in every nook and corner of our surroundings. Of these, certain microorganisms, such as bacteria, have a nucleoid but no membrane-bound organelles in their cytoplasm. Such organisms are called prokaryotes. Prokaryotes are smaller and multiply more rapidly than eukaryotes. All members of kingdom Monera, including bacteria, blue-green algae, and pleuropneumonia-like organisms or mycoplasma, are prokaryotes. And they can be rod-shaped, spherical, spiral-shaped, or comma-shaped. Microscopic studies reveal that most prokaryotes, except for a few such as mycoplasma and thermoplasma, possess a cell envelope, a complex, tightly bound, three-layered chemical structure consisting of the outermost glycocalyx, followed by the cell wall and then the plasma membrane. The layers together act as a single protective unit, though each has its distinct function. The glycocalyx, for instance, helps prokaryotes such as bacteria to resist antibiotics when they invade host cells. Did you know that the glycocalyx differs in thickness and composition among different prokaryotes? In some, it occurs as a thick and tough sheath called a capsule, while in others it may be a loose sheath called a slime layer. The glycocalyx is followed by a cell wall, which determines the shape of the cell and provides structural support, thereby preventing the cell from collapsing or bursting. Interestingly, the cell wall structure differs in different prokaryotes. In certain prokaryotes, such as Pseudomonas, the cell wall is composed of a thin inner layer of peptidoglycan and an outer membrane of lipopolysaccharide and lipoprotein. When such prokaryotes are subjected to the Gram staining procedure, they do not take up the Gram stain, but appear red or pink when counterstains such as safranine are added. Hence, they are called Gram-negative bacteria. On the other hand, the cell wall of prokaryotes such as Bacillus anthracis, composed of a dense layer of peptidoglycan and teichoic acids, turns violet when subjected to the Gram staining procedure. Hence, Such prokaryotes are called gram-positive. The cell wall is followed by the semi-permeable plasma membrane, which controls the entry and exit of small molecules like food particles and ions like chloride, sodium, and bicarbonates. Often, the plasma membrane extends into the cell, forming the mesosome, a membranous structure seen in the form of vesicles, tubules, and lamellae. Incidentally, the presence of the mesosome is a characteristic feature of prokaryotes. Mesosomes help in cell wall formation, respiration, secretion processes, expansion of the surface area of the plasma membrane and enzymatic content, DNA replication, and distribution to daughter cells. Some prokaryotes, such as blue-green algae, also have membranous extensions called photosynthetic lamellae that contain photosynthetic pigments such as chlorophyll. Prokaryotes also have extensions from their cell walls. For instance, motile bacterial cells have a thin filamentous extension from their cell wall called a flagellum, which consists of a filament, hook, and a basal body, with the filament being the longest portion. Motile as well as non-motile prokaryotes also possess pili and fimbria, surface structures that attach the prokaryotes to rocks in streams and also to host tissues. While pili are elongated tubular structures made up of a special protein called pilin, fimbria are tiny bristle-like fibers sprouting out of the cell. However, these structures don't play any role in motility. While pili and fimbria are present outside the cell envelope, the inside is filled with cytoplasm that hosts the genomic DNA, a single chromosome made up of circular DNA. Many prokaryotes also have small circular DNA called plasmids outside their genomic DNA, which confer unique enotypic characteristics such as resistance to antibiotics. The cytoplasm also contains ribosomes measuring about 15 microns by 20 microns in size, and consisting of two subunits, 50S and 30S, which together form the 70S ribosome. Here, S, or Svedberg's unit, stands for the sedimentation coefficient. It indirectly is a measure of the density and size of ribosomes. These ribosomes attach to a single messenger RNA, a type of RNA that aids in protein synthesis. forming a chain called a polyribosome or polysome, and ultimately translate messenger RNA into proteins. Apart from the ribosomes, cytoplasm also contains inclusion bodies such as phosphate granules, synophycean granules, and glycogen granules, which are used for storing reserve materials. These bodies are not bounded by any membrane system and lie free in the cytoplasm. Certain prokaryotes such as blue-green, purple, and green photosynthetic bacteria also have other inclusion bodies such as gas vacuoles, which help them to float in water. Prokaryotes, thus, are primitive organisms that possess a simple cell structure. Yet, they are the only organisms to thrive in almost all habitats, including underground caves and deep oceans.